Okay, so it's noon sharp here, and welcome everyone to today American Physiological Society webinar entitled Neural Network Inspired Model for Understanding Metabolism Behavior Interaction. My name is Lizzie Janes Cardoso. I'm the past chair of the American Physiological Society Endocrinology and Metabolism section, and I will be serving as your moderator today. In today's sections, we have Dr. Lee G of Script Research, who will discuss the use of a time lag linear mix model and temporal relationship among metabolic variables to provide a precise view of metabolisms in animal models. Before we started, a few housekeeping notes out of respect of our speaker. We ask you to refrain from photo or video. Taking photos or without the person taking photo of another person presentation is prohibited. Please note that at the end of this presentation, we'll have available the talk so you can watch it back or in the link on the registration website. I'll be happy to take any questions that you have if you enter in the Q&A function button on the bottom of your screen. And before I introduce our amazing speaker, I would like to thank our sponsor today, Sable System, for their support to the American Physiological Society and the mission to advance metabolic physiology to better understand human health as well disease. Before we begin, we will launch a very brief poll to learn more about your metabolic research. I think there are only 3 very short questions. If you can please answer those so we can better understand how to create content for your research. Yes, so now it's really my pleasure to introduce our amazing speaker, Dr. Lee Ji, who is a professor and an investigator of the Howard Hughes Medical Institute. He is the N. Paul Wither Endowed Chair in the Department of Neuroscience at Script Research. Dr. G received his PhD at Harvard University, and later on he did his postdoctoral studies at Stanford and MIT. In his lab, they studied neuroscience, the molecular mechanism, and chemical biology, and how they talk and how the central nervous system talk to the peripheral tissue to keep us healthy. So please join me in welcoming Dr. G. Lee. Dr. G, the floor is yours.
All right. Can you hear me well?
Yes, we can hear you well. We can see you well. And if you want to upload your slide.
Oh, can you see my slide? So it's already shared.
Yeah, I cannot see your slides. Jay, can you see slides? Yeah, I cannot see your slides.
I do.
Let me reshare it.
Please bring it up. Yep. Thank you.
Can you see the slides now?
I think it's coming. We'll wait 1 second. Is said share screen? Yes, now we can see well and we can hear you well. The floor is yours.
All right. Thank you, Lizzie, for the introduction. And I want to also thank the APS for this opportunity, and also thank Sable Systems for sponsoring this webinar. Let me see whether I can on my slides. All right. So I want to start with today's story, actually, how we come to this perspective of generating these new tools for analyzing metabolic behavior interactions, largely through analyzing the metabolic cage data. So I guess I will start with some of the biological questions. I guess we are a lab of neuroscience, and I guess one of the driving factors that why we are interested in metabolism and the interaction between metabolism and neuroscience is because this general notion that the body is a main metabolic support and for our nervous system. It's well known that during evolution, there's a pretty good linear size correlation between an animal's brain versus its body. The idea that although a more bigger and a more complex the brain will give you a lot of advantage, but this is fundamentally constrained by the metabolic support from the body. So this famous plot can also be plotted in a relationship between the body metabolic rate and then the brain metabolic rate, that you will see an even better linear correlation across the animal kingdom. Basically, the size of the brain and the metabolism of the brain is really constrained and dictated by the size of the body. So my lab is generally very interested in understanding how the brain and body work together to support this both neuronal function as well as the metabolic fitness. This is the analogy of studying, the energy supply of a computer and trying to understand some of the guiding principles of how the biological system actually integrate the neuronal control and the energy metabolism. It's some kind of ultimate goal of my lab's research. And one of the models we are very interested in using to study this body-brain interaction is using cold exposure. So in this famous plot, if you actually segregate the warm-blooded animal versus the cold-blooded animal, you see actually this linear relationship between the brain and body actually segregate into two different groups. So it is well noted that by developing, endothermia, basically to maintain a higher body temperature than the environment, that allows the higher metabolism, actually allows a more potent neuronal function, but it also comes with a cost, right? As many of you we know, that animals, especially small animals, when the ambient temperature is low, they actually have tremendous metabolic stress in order to maintain their body temperature at this higher temperature, higher level compared to the ambient environment. So in rodents, this is actually very well documented and very well studied. For example, people know that essentially all the mammals has to spend a substantial amount of their daily energy expenditure to maintain a body temperature. And in small mammals like lab mouse, like for example, if you put them in 4 degrees, they actually will double their energy expenditure in order to meet that, increase the demand of a cold temperature. And this is also usually accompanied by double their energy intake. So the mice will basically eat twice as much and then spend twice as much to maintain the body temperature. But this also, create a dilemma, right? Because you could, under cold exposure, as many of you probably know, a large part of the energy expenditure in mice, almost half of the energy expenditure in 4 degrees, is designated to their brown adipose tissue to generate heat through a process called the thermogenesis. Actually, in doing so, the body also shut down a lot of the so-called non-essential activities. So, for example, the mice will move less, they will explore less, they almost into a kind of a near sleep. Sometimes they will even enter torpor to preserve energy. So this create a behavior dilemma that if you preserve the energy too much, basically you will enter a forever coma that you never get enough energy replenished. So at the same time of saving energy, the mice also have to increase their food-seeking behavior in order to replenish all the energy they spend. So we are using this model as a way to understand how the mice actually adapt both metabolically and behaviorally in response to a cold challenge. So this work has been published a couple of years ago led by a very talented former postdoc, Niraj Lai, who now is a PI at the UPenn and the Monell Institute. So the key question of this study is how does the brain actually control the behavior adaptation in the cold to meet both to conserve energy as well as to increase the energy intake. So the very first experiment we did is actually in collaboration with Alex Banks' lab, who also gave, I think, an APS seminar not long ago. So Alex's lab really are specialized in using metabolic cages. They have a very sophisticated stable Promethean system that they actually can precisely measure energy intake and energy expenditure in lab animals. So we first repeat the experiments that has been described in the literature many, many times, that it's well known that if you put a mice in 4 degrees, they can actually happily live there for a long time. What happened is that they will double their energy expenditure when they were exposed to the cold, and then they also double their food intake. Again, this data confirmed what we already know from many previous literature and knowledges. What's really interesting is that other than taking this 12-hour average food intake or energy expenditure across a period of time, if we actually now taking advantage of this Promethean system has this higher temporal resolution of have this kind of more detailed data and look at the first couple hours after cold exposure, we see something very interesting. For example, you can see two lines here. The green line is energy expenditure of these mice after they were exposed to 4 degrees. You will see almost their energy expenditure immediately increase after the mice was put in 4-degree ambient temperature. However, this increase of food intake does not happen immediately. To some extent, you will see almost there's a couple hours of delay before the food intake actually increased, although this was reflected on the average level across many hours. There's almost an initial decrease of food intake, largely due to the stress that mice experience when you change their body temperature, when you change the ambient temperature to 4 degrees. It's only after this 4 to 5 hours where we define as a cold-induced energy deficit, because you can actually calculate the energy expenditure and intake. That's where we believe the energy homeostasis is severely perturbed, that you actually have an energy deficit. This is where the mice begin to eat. So keep this important time point in mind, because we will get back to this time point in some of the later talks. So also we begin to look at the video recordings of the behavior of these animals. On the left is the very typical overnight fasting experiment. For example, if you put a mice, food restrict them for 16 hours, that's how they eat. And on the right is when the mice are actually put in a cold, 4-degree cold environment, and that's how they eat. You will clearly see their behaviors are very different. In a fasted animal, they actually very active foraging behavior. They go after the food pellet almost continuously, whereas in the cold, the mice actually spend most of the time, hiding in the corner. That we believe this is a strategy to conserve energy and minimize movement and the brain activity. But they do come out once in a while, having this kind of concentrated bowed movement to take a large bite before they return to the corner to continue to shiver and then rest. So we think, despite these mice actually almost eat the same amount in this first, an hour after cold exposure or food exposure after food restriction, they reflect a very different behavior strategies. Then we can also quantify the behavior response in the cold. We consider these mice spend more than 90% of their time in a resting state, shivering, and basically it's a way to conserve energy of so-called non-essential energy expenditure. And then they spend about 4% of the time doing foraging and food-seeking behaviors. So to understand the neural mechanism behind this behavior strategy, we begin to do whole brain CFOS profiling through brain clearing and light sheet imaging. And very interestingly, we identified a very small brain region in the ventral medial part of the thalamus that is a very poorly understood and poorly characterized the brain area, called the Xi5 nucleus. Another reason this region caught our eyes is because in the literature, people have lesioned this area of the thalamus in hibernating hamster, and then they show the hamster actually will have a deficit in terms of the overwinter behavior response. So what we see very interestingly is, although if you put a mice in very acute 15 minutes cold exposure at 4 degrees, you do not see activation of this brain region as you see in the left. However, if you prolong the cold exposure to enter this energy deficit state that we estimate happened between 4 to 6 hours, so then you will see this prolonged cold exposure and energy deficit begin to activate this small area of the Xi5 nucleus. Then we went on to use in vivo calcium imaging through a fiber photometry in these behaving animals to record neural activity of this Xi5 nucleus. Before we measure Xi5, we actually measure a canonical feeding center, the AGRP neurons in the arcuate nucleus of hypothalamus, that many of you probably have heard of. So this is, again, very well known in the literature that in fasting-induced feeding, the AGRP neuron will have a very characteristic job of their activity every time when the mice actually approach food and then start feeding behavior, as you see on the left side of the fibrophotometry trace. On the right-hand side, to our surprise, that this canonical feeding center did not show this, feeding correlated activity drop or increase when the mice are actually eating in the cold. Basically, this cold-induced feeding bout is not associated with this stereotypical AGRP activity. But when we actually measure the xiphoid nucleus activity, we begin to see a very time-locked response of xiphoid neural activity right before the mouse initiate the feeding bout. But this is also not the case when the mice are actually eating under room temperature or just after fasting refeeding, suggesting that the neural activity of xiphoid is selectively associated with cold-induced feeding and energy expenditure with induced feeding. And then subsequently, we also did bidirectional either chemogenetics activation or chemogenetic inhibition. We'll see basically when you activate those neurons, the mice will eat more in the cold. When you inhibit these neurons, the mice will eat less in the cold. And you can also do so through a more precise or faster optogenetic manipulation. Not only you will see increased food intake in these mice when you are in the cold, when you stimulate the xiphoid nucleus, or you can actually behaviorally observe this kind of behavior switch going from a shivering, hiding state to a almost near arousal type of food, exploration behavior transient. And we also confirmed that neither the activation of xiphi or the inhibition affects fasting-induced feeding. So the canonical, this is a part of the mechanism kind of in parallel to the canonical fasting-induced feeding pathway that largely through AGRP and then related hypothalamus circuit. So this is just a quick summary of why we begin to develop our tools and then kind of get into the main story of the talk. So by looking at the cold-induced feeding and then looking at this high temporal resolution behavior metabolic cage data, we identify this kind of delayed response of food intake for the mice in response to the cold. But this promotes a bigger question that so in this particular study, right, we were able to manually pick energy expenditure and food intake, and then leverage this high temporal resolution data to look at this raw data instead of using the 12-hour average to find this very interesting temporal delay and the very interesting time window to study this interesting interaction between behavior and metabolism. But how do we generalize that? It's not possible for everybody just to take the raw metabolic cage data and then look at them minute to minute and hour by hour to look for such relationship. So this is motivate us to ask, can we develop a method to actually unbiasedly detect temporal interactions between physiology and animal behaviors? So this is a work led by a very talented PhD student, Han Bing-shen, who is also on the panel here. And I think later in the Q&A section, we'll have opportunity to actually directly talk to her. So Han Bing's question is that, can we develop a mathematical, I think there is a common, is there some echo issue in the?
No, we can hear you well.
Oh, okay. So Han Bing's question is, can we develop a computation method and essentially a software to allow us to actually unbiasedly look at all the potential temporal interactions between a particular metabolic parameter and the behavior parameter? So in the next, for the rest of the talk, I will focus on this part of the story. So let's first look at how people typically look at the metabolic cage data, right? So I think many of you are very familiar with this kind of, the standard way is that people take several days of metabolic cage recording. Then you have multiple parameters that in your hand, like including VO2, VCO2, and you have calculate energy expenditure, RER, food intake, water intake. You will have some locomotion effects in there. And I think later we will add more recording parameters. But overall, I think these 8 are very typical metabolic parameters that you will get from your metabolic cage data. And despite this, it is a continuous recording over 48 hours or 24 hours or even longer. And then the original sampling rate of these parameters, for example, in a Prometheus system, it's actually really high. Like you can get near 1 hertz, or this actually the raw machine is sampling them at a couple seconds. But most of the time when people get those data, right, you compress them into a 12-hour time window, and you take the means and you compare the means across different groups that usually people use to assess whether you have a phenotype, whether this is a phenotype due to a treatment, whether you change those mean values, or this is due to a genetic genotype between different groups that you're trying to understand are there mean difference. But we will argue that, based on what we show you, between the interaction of energy expenditure and food intake, there are many of those more subtle but highly significant temporal interactions that are happening at a minute or the hour time scale, that when you actually average those data by day or night, you actually lose all those, very interesting temporal interactions, and especially they are pairwise in the relationship. So you can imagine that in this, the code-induced feeding story, I told you about the interaction between energy expenditure and the food intake. But you will also imagine that, for all the parameters that we record, there may be some kind of hidden or latent interaction between energy expenditure and glucose and food intake and RER. And those are the things that I think people already talk about. Like, for example, we know RER will change with, food intake. We know movement will likely associate with food intake. Those are the things I think we talk about when we look at the raw data in the metabolic cage and in an anecdotal way. But I will argue that currently, as a field, we don't quantitatively or in a standardized way to actually quantify and compare those relationships. And that is kind of one of our initial motivation to look at those interactions. So how do we do this? As a neuroscience lab, and one thing that actually we do a lot in modern neuroscience, especially circuit and systems neuroscience level, we do look at the temporal interactions with very rapid signals and their interactions. For example, the neuroscientists record from different brain regions with very high temporal resolution electrical or calcium imaging based recording. So this is one example that we just pull out from the literature. This is the single unit recording from multiple neurons in the neocortex of a mouse. So each row of those signals corresponding to a neural activity of a single cell in a single neuron across the in the mouse cortex. So you will see that the neurons actually they spike at a different time point. But there is mathematical models. Basically, you can infer their connectivity and then a causal relationship of this network simply by assuming that, hey, if neuron 1 spike at a particular moment, and if another neuron spike immediately after this first neuron, and if across a long period of time this relationship persists, then you can infer that maybe neuron 1 and neuron 3 are actually now connected. And also, if you look at who get activated first and then the subsequent follow-up activity, you can also infer who's upstream of who. And people do this through a very well-established mathematical model called a generalized linear model, or GLM, to infer this so-called microcircuit relationship based on neural activity data. So what Hanbin come up with the idea is that if you actually plot the metabolic cage data as individual parameters and lay them, stack them on top of each other, and it looks like a raster plot like this. Essentially, you can imagine that you can treat each of the metabolic parameter you record from metabolic cage as a neuron. And since they have up and downs across a long period of time, you can actually infer this temporal relationship using a very similar algorithm that when we used to use, basically it's a GLM-based algorithm that we use to infer relationship of a neural circuit. And this is where we begin to use this kind of GLM to understand the temporal relationship between different metabolic parameters that record from the same animals. So let me just dive in, slightly deeper of exactly what GLM is doing. I promise I won't get into too much of the math. So in general, if you think about a pair of metabolic parameter or a pair of neurons, you can treat basically one of them as a so-called stimulus and the other as the response, right? So if you assume every response, then here this is color-coded in red, and then we consider the response at any given time t, as we designate them as a response y. And if we assume in every given moment t, the y is a linear combination product of the stimulus x, so then we can come up with a model basically trying to describe y as a linear relationship between a specific parameter of x plus a Gaussian noise. So one specific example in neuroscience is like, for example, if you can use different neural stimulation and recall from different cell types, then you will see, for example, if you use GLM to output their coefficient over here, we name it as a temporal filter. This is an important definition that will keep come to, terminology will keep come back to in some of the later slides. So for example, in a neuroscience study, we can give the system a stimulation and see which neuron actually responds to this stimulation, and their responsiveness to a particular stimulation is being output or being quantified as the temporal filter. So in this particular example, as you can see, the purple neuron and the purple neuron is responds to its own target stimulation. Therefore, they have a positive temporal filter, whereas the green neuron and or the black neurons, they don't respond to a particular stimuli, so they will show a flat temporal filter, shows there's no correlation in response to the stimulus. So in a typical neuroscience setting, we use the kind of the parental generalized linear filter to look for this kind of microcircuit relationship. But in the mouse metabolic cage study, because we are dealing with a much bigger variation across individuals, so we actually modify this model. What we are actually using is a so-called linear mixed model. I think the only difference here, you can, oversimplify term, I guess the only thing we add is this random effect on top of the Gaussian noise to actually account for individual variations across individual animals. Now, again, we go back to our favorite collaborator, Alex Banks' lab. So we got a very high quality Promethean recording data from 24 mice. This is a very large N number. This is high quality data, very well calibrated, recorded over 48 hours. The sampling rate is downsampled to 3 minutes for the convenience of our analysis. There are 8 parameters, as I mentioned, in this data set from VO2, VCO2, EE, RER, and food intake, water, directed locomotion, and all movements. So again, I want to give you a more specific example of what we do in this specific example. As I mentioned, like a reminder that we treat one parameter as stimulus, and then we treat another parameter as its potential response. So like, for example, we can, many of us know that, by looking at the raw metabolic cage data, we know feeding and drinking tend to happen close to each other in terms of their timing. In a typical, wild-type mice at night, when they drink, they tend to eat at the same time. So now if you look at the drink as an output, if you look at the drink as a response and use feeding as a stimulus, and you will get this temporal filter, shows you that basically every time there is a feeding, it's very likely in the next 0.4 hours, there was very likely there will be a drinking event also happening. So suggest that basically this relationship is being characterized as a positive temporal filter. And this is confirmed by if you actually align the raw data together. The red line here is the moment, every time there's a feeding onset, and you will see the likelihood you observe a water intake is very high right after a feeding event. So this is a formal and a mathematic description of what we anecdotally say, hey, feeding and drinking tend to happen together. And similarly, we also know, eating tend to increase energy expenditure. This is being described as a, feeding-induced thermogenesis. We can also, directly quantify and characterize this relationship as a positive temporal filter between feeding events and the energy expenditure. Again, we can align the raw data to show this is truly reflecting the ground truth of the animal behavior and the physiology. So now with these examples, since we have 8 parameters, you can see we can do this pairized analysis across all potential combinations. So the output of the model is essentially 56 pairs of interactions. And this is very difficult to look at and then very difficult to even manually analyze. So we actually use a pretty typical way to actually cluster them together, UMAP. Probably many of you are already using them for your single cell analysis, but we can use basically a similar dimension reduction analysis to reduce this 56 pairs of interaction into 3 types. In the next slide, I will describe what are the general 3 types. So, and many of you are familiar with metabolic cage. You probably already have this question now because not many of those parameters are truly independent of each other indeed. For example, the energy expenditure is actually calculated by a linear formulation between VO2 and VCO2. So under those circumstances, basically the temporal filter will just look like a straight line at a time 0 because essentially mathematically you are looking at 2 dependent variables, right? So you will see this between EE and VO2 and likely between VO2, VCO2 and EE as well. So basically we almost consider this dependent relationship as a mathematical artifact that basically if the 2 parameters, they are linear combined, the linear product of the other, you will see this so-called dependent relationship. That doesn't really tell you much of the underlying biology. However, there are other things that are biologically way more meaningful. For example, as I mentioned, that people describe the feeding-induced thermogenesis. And you can see this every time when the mice eat, there's actually a long-lasting effect on their energy expenditure. This is where this type of interaction, we call them the long-lasting effects. So this is, I use a pie plot to see that all the potential the interactions that fall into this category, for example, between RER and movement, between feed and RER, between feed and EE, this is fall into this category of a long-lasting impact of one versus the other. We also have something more intermediate in between. Basically, it's not immediately sharp as the dependent one. It's also not as long-lasting as the feed to EE. So those are the examples. We call them intermediate. For example, the impact of feeding on a RER, it lasts for lasts for about half an hour, but not as long as the others. And then you see more of the dynamics and fluctuation up and down across the shorter time scale. And then again, we have many pairs like this across the whole spectrum. So we also now run this algorithm across multiple data sets from Alex Banks' lab, for example. The original method development is from this 24 mice, 2-day recordings, 3-minute sampling rate data set. We call them data set 1. We now benchmark this across many different data sets that was recorded at a different time, different year, different cohorts of mice, different age, and maybe even potentially different genotype. So we have data set 2 coming from 16 mice, 6-day recording. We also have data set 3 is our smallest data set, 10 mice, probably closer to most of your daily experiment. This is recorded from 3 days. And you can see that all the 56 pairs that actually remain quite stable, even when we analyze this relationship across different data sets. And then the UMAP also remains very consistent. It gives us these 3 types of interactions, the dependent, intermediate, and then long relationship. And then we can summarize this into interaction pie chart. And then on the right-hand side is just some of the specific temporal filters we pull out to show each row represent a biological cohort that will see basically both the overall landscape as well as the individual temporal filters remain highly reproducible and consistent across different data sets that collected at a different time and collect from different animals. So one of the benchmark examples. So basically, what happened is we now have 48 stable benchmark pairs that shows statistically significant difference between the day and night cycles. As we know, the circadian rhythm has a huge impact of mouse metabolism and behavior. So we do detect there are 48 stable pairs that shows consistent difference across multiple mouse cohorts. So we call them the 48 benchmark pairs that will allow us to actually quality control of any of the new cohorts that you can basically, any de novo generated mouse data, you can use their day and night difference as an internal quality control to see how well the algorithm will actually perform on your own data set. This is one example of looking at one pair of relationship consistently show highly significant and reliable and biologically meaningful difference between day and night. So in here, there's two lines here. The yellow line shows the temporal filter during the daytime, and then while the gray line shows the temporal filter during the nighttime. And then the star denote that there's a statistical difference, and then a false discovery rate below 0.5 under those two conditions. I don't think I need to explain what RER is for this audience, but in general, this is basically quantifying the fuel utilization between a carbohydrate versus lipid. So what this difference show you is, regardless whether it's daytime or nighttime, feeding will transiently increase RER in animals. As you see, there's a positive bump of the temporal filter in relationship between feeding and RER. What's really interesting is that basically this temporal filter tells you during the daytime, feeding has a much stronger impact on RER compared to nighttime feeding. Basically, during the daytime, when the baseline RER is low, even when the mice wake up and take a small bite, this will have a much bigger impact on their RER compared to their feeding events during the night. Again, so right now we established the baseline of the method. Basically, we have a stable algorithm that can generate highly reproducible 48 benchmark pairs that shows day and night difference across multiple data sets. And we can quantify and then visualize those interactions through this pie chart. And so to both get a global view of the interaction map as well as individual and interpret and then quantify individual interactions. So we begin to now use this for some of the more discovery or exploratory applications. So one of the newer data set we got from Banks lab is this now 10 mice and almost near 1 month of high resolution recording. So the Alex's lab put these mice into multiple well-established metabolic challenge from 30-degree warm thermoneutral exposure to overnight fasting to 4-degree cold exposure, high-fat diet treatment, and we see how those relationships will change under those conditions. And very interestingly, on top of the 8 parameters that we typically get from a metabolic cage, Alex also add both telemetry measurement of the core body temperature as well as the aorta measurement of the circulating glucose level, continuous glucose measurement to allow us to get a more rich data set and to model. Also tested whether how the method will actually hold up when we actually now have 10 parameters instead of 8. So the first application is we want to look at the metabolic interactions under 2 energy deficit states. So we know that both cold exposure and fasting can induce energy deficit state through different mechanisms, one through increased energy expenditure, the other through restricted energy intake. So we wonder, the first thing first, we did a more traditional analysis. So although I put this into a range, but this is just literally comparing the mean value across these 10 different parameters. So basically, the star just means that from a t-test of whether this reaches significance or not. And again, this is not new information. We know under cold exposure, energy expenditure go up, VO2, VCO2 go up, the body temperature slightly decrease, and whereas during fasting, your energy expenditure go down, VO2, VCO2 go down, the body temperature also goes down, but your body glucose, the blood glucose go down. Whereas in the cold, you actually have almost a slight increase of blood glucose. What's really interesting is now if you begin to plot the global interactions across these 10 parameters under those 2 different ways of generating achieving energy deficit, you see a much stronger interaction between RER and core body temperature under the cold conditions, whereas you don't see the same interaction coming out of body temperature RER in the fasting. I think the main driving interaction changes are really coming from energy expenditure, VO2 and VCO2 under fasting conditions. And that also allows us to detect the supple individual pairs. So first thing first, I want to go back to the original story where we, led us here. Remember I told you energy expenditure can drive feeding more significantly in the cold. And the first of the thing we want to test our pipeline is whether we can actually quantify and describe this phenomenon. And indeed, if you look at energy expenditure and then the temporal relationship with feeding, you can actually now detect a significant increase of EE-derived feeding under 4-degree conditions and reach statistical significance, confirming of our manual finding that under cold conditions, the energy expenditure now has a stronger effect on driving feeding. But what are some of unknown or the new things that we discovered from these interactions? One of the very interesting example we found is the body temperature, how body temperature actually have an impact on movement or predict movement. So under room temperature was showing here in the black line, and we know at the baseline, basically the body temperature is actually a very strong predictor of movement. So as you can see here, there's a very positive temporal filter. Under 23 degrees, every time body temperature increase, very likely that predicts the next moment you have a movement. However, this relationship was largely almost abolished when the mice are in the cold. So basically, because they largely choose to hide and shiver, this kind of body temperature prediction on ambient movement has been largely diminished, which is not the case when actually the mice are being fasted. In the fasting mice, that despite they are actually also in energy deficits, since they don't choose this kind of shivering and energy conservation strategy, they remain to have this very strong correlation between their body temperature and their ambient movement. Another very interesting example is the body temperature and glucose relationship. But both in the room temperature and in cold, the body temperature has a pretty strong prediction value to their to their blood glucose level. However, this relationship is completely reversed, almost changed during the fasting state. For example, if you overnight fasted animals, now the body temperature has a much shorter but sharper prediction value towards the blood glucose compared to either room temperature or fasting. Another very interesting application we begin to test is how high-fat diet actually change the temporal relationship across all these parameters. So we actually have two different time points. The first time point is when the mice are only exposed to high-fat diet for between day 1 and day 2, or they have been exposed to the high-fat diet for 8 to 9 days. And again, looking at the average plot, if you look at the mean value across data between day and night, they actually look quite similar. I don't think we can reliably detect any difference between the 2-day versus 9-day high-fat diet in those early stage of diet-induced obesity. However, if you begin to look at the interaction map, some very interesting patterns start to emerge. For example, we see in the early stage of high-fat feeding, a lot of the perturbation are actually concentrated in energy expenditure and VO2 and VCO2, whereas at the later phase, the feeding behavior and even drinking itself begin to get perturbed with prolonged high-fat diet treatment. We can also look at the individual pairs. For example, as I mentioned from the very beginning, it is well known that in wild-type healthy animals, the feed and drink tend to happen together. As you can see in the black line here, so this is the control conditions that the feeding and drinking tend to happen together. So as showing as a very positive temporal filter. As you can see that basically as the mice were put on high-fat diet, even from day 1 to 2, you have a diminished correlation between feed and drink. And then when the mice are actually exposed to high-fat diet for more than 8 days, this becomes even more severe that basically this strong correlation between feeding coordination has been kind of heavily disrupted by high-fat diet. Another example is that under normal conditions, the energy expenditure is a pretty clear predictor of feeding, as we show in the cold scenario as well. However, these relationships also get rapidly disrupted by high-fat diet. So even with 1 to 2 days and become more severe after 8 to 9 days, that the energy expenditure driven the prediction of feeding become weaker and weaker and largely diminished. We are still trying to interpret the result, but one potential interpretation is that the mice go from, after prolonged high-fat diet exposure, their feeding kind of go from a more homeostatic regulated feeding to a more hedonic driven feeding. Another ongoing work of some of our still kind of very early stage work is we begin to look at different anti-obesity drugs and how they actually affects this relationship. So we actually use 2 different cohorts of mice exposed to either semaglutide, kind of the most well-known GLP-1 agonist versus some of the newer experimental dual agonists towards GLP-1 as well as glucagon, and then look at their relationships. Again, by using traditional average comparison, these 2 drugs are actually leading to very similar overall metabolic changes. For example, they reduce food intake. They actually cause a decrease of energy expenditure, slight decrease of movement. So this is largely very similar between these 2 conditions. Again, this is still very early, so we are not 100% definitive yet, but some of our early analysis suggests that some interesting difference between those 2 drugs. For example, one of the more distinct pattern of changes perturbed by survodutide is that we see a much bigger perturbation between the movement. As you can see here, basically from the green blot, that AM and DM, these are the ambient movement and all the directed movement and ambient and all movement has appear to have a much stronger impact due to survodutide treatment from the interaction between the movement to energy expenditure. So a quick summary, basically we develop this algorithm and then a software to allow us to unbiasedly profile those kind of multi-directional temporal specific interactions between all the common metabolic parameters that we recall from a metabolic cage, and then we are leveraging this, the high temporal resolution data from the raw metabolic cage data, and is largely compressed and lost during a conventional analysis that we believe we can extract out of very interesting temporal information and then infer the physiology and behavior underlying biology of many of the different, both metabolic treatments and potentially different genotype, different drugs, and then could be, allow us to detect a lot of previously underappreciated biology. So with that, I want to end with talk with a kind of overview of my lab. I think, we are having this unique combination of 2 development and biological discovery within the lab. I think our overall goal is to both, develop new tools and then using this to establish new biological models to understand the metabolic communication between the brain and the body. And we hope this will allow us to discover the general principles guiding this brain-body talk. And eventually, hopefully this information will allow us to understand both the good and bad physiological and pathophysiological adaptations across those interactions. With that, I want to thank my lab, the people who did all the work, especially a shout out to Hanbing, who did all the computation work, and our collaborators, and some of the already mentioned Alex Bank, but a lot of our computation and the modeling work was led by our collaborator Mikhail Ao at UCSD, who is truly a world expert on generalized linear model, and also my collaborator at Scripps and Kennedy and many others, and our funding source as well. From there, I guess I'm happy to take questions.
Thank you so much, Dr. G. That was an amazing talk. Thank you again. And I do want to welcome Hanbin Shen. As you said, she's the graduate student that helped a lot putting together this data. So she'll be happy to take any questions that you may have. And so I'll start with question about the GLP-1. So we know that the weight loss effect of the combinations are significantly bigger than the monoagonists, the semaglutide. So if I understood right, when you use the combination of the glucagon agonist plus GLP-1, you saw more energy expenditure with movement compared to the monoagonist. Is that correct? And if that is true, can that explain the bigger weight loss effect that we see with the combinations?
I guess that's what we're trying to hint. This is, I think, this analysis doesn't really tell you this quantitatively more. I think what we can say is the movement have a bigger impact on the movement itself, rather than has a amplitude of. It's not the absolute amount of energy expenditure. I think it's the changes on energy expenditure it become more significant due to the movement.
Because you didn't see any difference between food intake among those two, right?
Not necessarily. I think even if we directly measure the energy expenditure, there is no absolute amount increase.
Right.
It's the increase is more, correlate. It's driven by the movement itself rather than the absolute amount increase.
I see. I see. Okay, so we'll take a question. So please, if you have any, you can type in the Q&A button. So Tyler have a question, and he asks, with regards to your calculation caloric deficit, which equation are you using to calculate the kilocalories available for metabolism? And he further asks, will there be any difference in GI absorption with cold that could potentially hamper the kilocalories available for energy expenditure? So when it's cold weather, do we absorb the same? So we eat more. Now the question is, do we absorb equally more? Great question.
Yeah, no, we cannot directly infer that. Like I said, I guess it's probably more accurate to call this the apparent deficit rather than we are not inferring this is the real deficit because, again, largely this is a computation method trying to interpret the data from the metabolic cage. I think this is the kind of the face value of deficit rather than we cannot, with this kind of data, we cannot go in to infer absorption, or there are things that we cannot speculate the things we don't have information on. Yeah.
Okay. So we have another question from Dr. Romero. Excellent and stimulating talk. Is there any way to know if there any of those relationships are association or cause-effect interaction?
Again, I don't think we can draw causal statements here. This is the statistically the math doesn't allow us to do that. This is basically a hypothesis generating profile. It's the same thing like any other high throughput method. It's give you the idea that you have to test the causal relationship biologically, just like any omic based method. I guess maybe Hanbin can comment on the causal versus correlation statement.
Yeah, sure. I can answer this question. So what we observe here is mainly we want to interpret how two variables are correlated each other in temporal domain. For example, if one variable change, does another variable respond immediately or respond with a delay? And this can give us very high biological insights. For example, after we eat, people would be curious, does our glucose increase immediately, or does our glucose rise with a delay, and whether this would be related, for example, obesity, high-fat diet. And for the causal parts, we still need to do experiments, like Li have mentioned, the energy expenditure and food intake in the cold exposure conditions. And Niraj, which is a postdoc, have done experiments kind of to infer from that observation, do experiments to validate some of the hypotheses. So you can regard our platform as an exploration tool and to find out some pairs of interest, and then we can ask some questions to do the follow-up causal experiment in mice or in any animals there.
Yeah, absolutely. So we have another question from Lee. What is the data collection interval? Additionally, if you are using a sparser data set, example, binder or average data, do you still observe similar results from the GLM analysis?
So the so-called raw data, which I think is still processed the data we got from Banks lab, is at a 1 minute interval. We downsampled it to 3 for again, otherwise the computation cost is very high. So we actually downsampled to 3 for our analysis. I think maybe SAPO can answer more theoretically how high their raw data collection can be. I believe it's much even higher than 1 minute interval. We have tried to downsample it further. For example, we have data that's sampled as 6 minutes or sometimes even worse. I think the GLM will still give you answer, but the major limitation is when you have a longer interval and you have sparse data or binned data, you drastically lose your statistical power. So I think that's what we experienced. Basically, when you have sparse sampled data, then your ability to actually detect a low FDR or have a significant value, you basically lose statistical power to detect any difference in your cross-group comparison. All right, Kambing, I think that's the case.
Yeah. Yeah, I think what Lee most say is correct. And also I want to add more about like the interval things. The reason we choose like 3 minutes is because we want to keep the old data sets in our analysis pipeline consistent. And if we use, for example, more high frequency data, we maybe can find more pattern that cannot be captured by 3 minutes. So it's kind of a decision-making process. And 3 minutes, I feel, is both keep a balance between biological finding and also the cost of the time for the model complexity. Yeah, and I also have done some preliminary things like for a long time ago, like I'm comparing 1 minute, 3 minutes, and 5 minutes. And the results are pretty similar because like maybe it's not like very big difference. Yeah, so I don't think that's a problem. And based on most people's like metabolic cage sampling rates, I feel like most of them are kind of 3 minutes, 5 minutes, 1 minute. So yeah.
Yeah, that's great. So we are not going to have time to answer all the questions, but please do write the question because Dr. Lee and Han Bin, they are going to answer each of those questions after the webinar. So one more. Related to the assumption under GLM and linear mixed model, are the mice single house? If not, do models account for potential correlation of outcome? Example, REA among cage mates.
Maybe I can.
Yeah, I think in those experiments, every mouse is a single house. I don't think you can house the multiple mice in a single metabolic cage.
Right, right. So, and we have another one. Yes, excellent presentation. So a lot of good positive comment about your talk. That was amazing. How do you handle the common issues with cold exposure and high-fat diet feeding, increased humidity, increased food spilling to ensure they don't drive the effect that we see? So food spilling with high-fat diet.
I actually don't have a very good answer for this. I think this probably would be question to refer to Alex Banks or potentially Sable. They probably have much better answer for this. I do think this is in some of the early experiments, this is issue, but I think overall by now people have come up with very smart ways to probably not fully eliminate this caveat, but at least largely minimize this from the hardware side.
Right, right. Another question. For the anti-obesity drug comparison, where weight loss is dynamic, will it be more appropriate to capture interaction data at 2 discrete 24-hour time points like baseline and endpoint?
Yeah, we have done. So instead of doing like, week-long versus week-long data comparison, we have done like, basically truncated time window and then say the first 24 hours versus the last 24 hours. So we have done those experiments and there are some interesting observation there, but I think a general consideration is that I think a lot of maybe I didn't explicitly say that, I think a lot of our statistical power of this model is rely on the long recording period, right? So compared to a typical the neuroscience study, we have much smaller parameters. We don't really have 1,000 neurons to gain the power. Our statistical power coming from the longitudinal study. So when we actually, every time when we truncate this to a specific time window, then we worry about, we'll see difference, but then we lose the statistical power to actually to detect a statistically meaningful difference.
Very good. So this will be the last one. As I said, you still can write your question in the Q&A, and then we'll get to those. But this question, so one of the comments that I'm reading here, that they say that in Europe, many metabolic experiments have multiple mice in a cage. Huh? So apparently over there, they can have several mice in one cage. But this is a question. In the experiment where you look at the TV predicting locomotor activity, where you compare between 4 Celsius and fasting, do you remove the bouts of torpor, or are those what drive the correlation?
So at least we know in our data set, there's no mice actually went into torpor. I don't know how Banks Lab control for that, but we specifically asked about that.
Sorry for this question. So what is torpor? Remind me.
So I think it's there's energy deficits, especially with hunger and cold together. Some mice will enter this torpor state. It's essentially like a near sleep, but not sleep. It's like a hibernation-like state for rodents. But I think this is, again, depends on the gender. I think they are also like more likely to happen with female, and then they are likely to happen with specific light cycle. At least from the mice we are using that, I think the Banks Lab confirmed that at least within the data set we are using that. I don't think there is mice actually went to torpor.
Yeah, it will be nice to see sex difference in all these parameters, right? So as I said, many more questions. Unfortunately, we don't have the time to answer all of them, but I wanna thank again Dr. G for this amazing seminar. We truly enjoyed it. I learned a lot. And then I also wanna thank all the audience that take the time to be with us today. We really love to see you. Thank you for the questions and the great input in the activity. And again, we want to thank Saber Instrument for their supports to our activity this morning. And finally, I want to thank APS for organizing this webinar. Jacob Y. Jake, thank you so much for your help. And to all of you, good to see you again, and we'll see you soon. Thank you again. Bye-bye. Don't forget to fill the survey at the end of this activity.